and we need to, okay, good, thank you. And so we need to identify the injury site on skull. So we need to use a stereotactic surgery. Okay, later on I'm going to show you what it is. Okay, and we need what kind of tool to induce brain injury in animals. Okay, I'm going to briefly introduce to this. Okay, then open up the skull on a mice or mouse. Okay, use the scissors or scalpel, open up the skin or head scalpel. Now you can see that there. You can see this both picture here. There are a couple lines here, right? This is important for us as the mark allow us to do repeat injury in certain spots. Okay, we know each region, each lobe of the brain has particular function, right? So when we do perform experiment, we want to repeat the same condition, same trauma, injury in the certain spot of brain. Okay, this is good information for us. Okay, here is frontal suture, and this one in the middle line we know sagittal suture, right? And now in this one is occipital suture, and the close point. There are two close points. Here is a Berman and a lambda. Okay, there are 2 points. So this information can allow us to do experiment to make an injury repeated at some spot. Okay, that's the information for you. And this tool allow us to put the mice in and fix them. It's not going to move. However, I mean the reality not move on stage, but we can carry the stage according to this map. Okay, there's some scale, forward, backward, right, left. Okay, so we make a mark first. For example, I'm going to do an injury this spot. So we mark it first, then use this machine, move the stage and practice it before we make an impact. Okay, so when we get an impact later on, we are going to hit this spot. Okay, so we can repeat this kind of experiment from mouse 1 to mouse 2, mouse 5, and get data. Okay, so my lab. We got this one. We put the mice on over there, and they fix mice head over there. You can see that. And then next on, we are going to how to make an impact injury. Okay, this one we call them. This is a very simple one, weight drop. Okay, we got a weight here through the tube and drop it right and then fold. I mean, they will hit the brain. I hit the skull directly. Okay, so we need to try. We don't want to kill the mice animal right away. So we need to adjust the weight. We need to adjust the height. Okay, so we make right condition. The mice will survive, but get injury. So it's allow us to do some drug experiment for it. Okay, so that's we call them weight drop. It's more relative to our for sports mediated injury or or bicycle or motorbike induced head injury is more relative to. Okay, but the impacts are huge, and it's not easy to define which region get the impact, get the insult. So it's not easy to analyze molecular mechanism in that area. Okay, species not pinpoint particular nerve. Okay, and the other one we call it CCI. Okay, is a control cortical impact injury. This it can be regulated by computer. Okay, so we can digitalize, tell you how fast all this needle, how long will stay in the tissue. Okay, and what's the speed, Okay, so we can calculate everything and hit certain parts. So it's repeatable. So every mice will receive same impact, same spot, and then allow us to do experiment. Okay. Now we go to next. That's, for example, we can adjust the velocity, we can adjust the depth, we can check the duration when you hit it, how long they will stay on the brain and restore. Okay. So use this method, we can repeat our experiment. And next one, also in the TMU, we got another one. This project was used to work with NIH. You know, Americans like to send their troops overseas to fight for war peace, right? So a lot of soldiers got injured, suffer from the blast, from explosion. And then when those veterans back to the United States, they got some syndrome. We call them blast-induced TBI. Okay, so we mimic this explosion. We build up the pipe. And then between there are two segments of pipe. Between them, we are going to put the plastic paper or film like this, put in there between the segments. And in the other end, we put a gas tank over there, make a pressure. Eventually, it's going to break on this plastic paper or glass, which make a huge sound. And the animal on another side. What happened? The explosion, and will induce the brain injury in the animals. And use this way, we're working on the mechanism, we're working on drug, which can reduce the for the treatment of brain injury. Okay, after those impact, explosion or hitting, what happened? You know, neuron with a cell body with an axon. What happened? The vibration will cut off or make a piece by piece or axon, right? That's an injury. After neuron injury, who were detected? Another very important component of cells, microglia in the neuron, which were detected. For the purpose is to, what happened is for regenerating or healing this injury. They will release a lot of cytokine, a lot of growth factor, which is toxic to neuron also. And also another thing is the BBB. You know, the BBB is a protection membrane, right? Blood-brain barrier, right? Which can protect the neuron. So what happened? A lot of white cells in the serum will infiltrate into the tissue, so make a secondary injury. Then, so in my previous study, we try use the antagonist to block secondary injury, not allowing, prohibit neutrophils, white cells, T cell, B cell go into the brain. Okay, let's see this one. Without the impact. Okay, that's a film. You can see the mice goes through this bridge freely, right? No hesitation. And after impact, you can see that slow. If their four limbs is not going to coordinate. Okay, and it spend a lot of time to complete this whole journey. And it stop a couple times. Those information is our data. Okay, and then later on we are going to give the mice after impact with the drug and see their recovery is good or bad. And according to those information, we will check this assessment table and give the numbers. So using this method to figure out our drug is working or not working, is improved or not improved. Okay, just demonstrate some methodology to evaluate therapeutic evaluation. Okay, next one. Everyone should know one thing. TBI not only affect brain, but also affect a lot of different organs, lung, kidney, liver. It has approved a lot. Okay, so you gotta aware that. So every time you remind my student, after we execute the TBI animal, later on we need to check internal organs with the infiltration or not. Okay, so when you patient suffer TBI, I mean medical doctor also need to check other organs got damaged or not every couple of days later. Okay. I spent probably 10 minutes, 20 minutes regarding my report. I wish I can make you more interesting on this subject. Okay. Other people how to approach to find a drug to cure a traumatic brain injury. Now we are going to shift this lecture. I think everyone recognize these slides on the left. Last week the professor demonstrated the two different systems in human body: digestive system, respiratory system, right? So there are a lot of different systems in our body, like a assembly line in the factory. They want to make a part of, they want to complete the product, right? By assembling different parts of product. Okay, so they need someone to organize them, to coordinate them, right? A team and a computer. Same to the human body. Everyone, I think okay, I remember when I teach, one of my favorite course is biochemistry. Everyone took this course before? I remember always the first chapter is tell you what is a buffer, right? Why is it so important? Buffer is the solution can resist pH changes. It tells you all the biochemistry, all the metabolite reaction need to working under certain condition. pH affected by oxygen in our blood, carbon dioxide, right? And also affected by temperature. So we need to maintain temperature at around 37 degrees for optimal enzymatic action, right? Another one need iron. So all the function, metabolite function in all those systems need to one control center to regulate those conditions or execute those chemical reactions, correct? Who? Neuron and the endocrine system. Endocrine system, they release protein hormone to regulate, to maintain a good condition for allow them to execute their bioactivity or chemical reaction, something like that. Neuron, they were used member potential, one of the member potential to deliver for contraction. Okay, so nerve system, endocrine system is a very important organs. The system to maintain homeostasis in human body. That's the reason why in our first lecture always start with these two systems in human biology course or anatomy course. And they will look at the cells. The system derived from single cell, right? Every cells, eukaryote cells always has a membrane, right? Why is a membrane? Membrane is lipid, okay, contains two parts, polar and non-polar, right? And in the membrane, you can see a different stuff in there, protein stuff, For example, here, protein and the receptor. Okay, protein and there is an ion channel. Okay, so many stuff in there, which give the cell particular markers, allow cells to receive the hormone or external message to communicate with environment, with the cells. Okay, and also with the contact with other cells. Also help cells to migrate, to move. So membrane embed a lot of different protein and modify things, allow cell with a lot of different activities. And this membrane, this protein in the membrane is not fixed. They are movable because membrane is not stationary stuff. They're like an ocean. Okay, and receptor or protein like a boat, they can move around. The reason why I mentioned that, I will tell you later. For example, a lot of patients, for example, breast cancer patient, they will tell you overexpressing certain receptors. And then certain those receptors will migrate, shift. Unfortunately, if those over expanding growth factor making the dimer connecting each other, what happened? They were activated and the general signal, and cell were out of control for growing and division and turn to the cancer. Okay, that's another idea. Last, the component lipids. I mean, the membrane is a lipid, polar, non-polar. The tail is non-polar. So it's bilayer, it's a fluid. This is a very important concept on this slide. When you see the non-polar tail here, it's very straight. In other words, they are very rigid. Under the cold condition, they are easy to freeze. When they get frozen, the protein is not going to move around, will affect cell activity. Okay, so during the winter, a lot of fish, they need to modify their membrane by modify on the tail, make curve or insert the coaxial inside, whole big molecule in the membrane, increase the space between the membrane here. So we drop the freezing point and maintain fluidity of the membrane. Okay, so change the components of tail part is very important for the cell, their life. Okay, that's a very important idea for you guys. Okay. Now here, the membrane protein I just told you before, the receptor protein, they got the recognition protein. What's a recognition protein? It's a marker protein. What's a marker? Tell you your identity of these cells. For example, stem cells, hematopoietic stem cells, right? We are going to bone marrow transplantation. We need to collect the bone marrow stem cells, hematopoietic stem cells, not other cells. So each type of cells with their unique markers, like a leukocyte CD4, CD8, right? They are particular markers. Another one, enzymatic protein, which can, for example, they can facilitate some protein to digest them, make a small fragment for ingest into the cell body. Another one, attachment or adhesion protein, for example, leukocytes, white cells, in order to respond to the injury or inflammation, they need to recruit into the injury site by those adhesion molecules, right? For movement and staying, to clean the pathogen or for healing of tissue. Another one, transport protein, which can get the nutrient, get the oxygen from external environment into the cells. Here, this is important part for our lecture for transport protein, iron, neuron transmitter, okay, calcium iron, something like that is very important for us. Okay, so the idea membrane separate environment and the cytosol. They need to have the they got this, it's just like cells. However, they still need to get communication between environment and the cell body, correct? Okay, so what kind of substance will get into the cells? Depends what property of the substance, polar or non-polar, and depends their size. Okay, depends what kind of receptor they will recognize. Okay, so we can divide into three types. Passive, we call it passive transport. Those kind of transport, they don't need the energy. They don't need to ATP. They can freely go into layer. For example, simple diffusion. What is simple diffusion? If the media has a certain gradient, it's easy to go through in there, right? From higher concentration to lower concentration. That's simple diffusion. What's a facilitated diffusion? They need to help what kind of molecule, protein to help it. Osmosis, osmosis is regulated by water gradient. There, I remember, okay, I should mention this one, okay, get in there. For example, like this one, if you are a nurse, you are withdraw blood from the patient or healthy individual for clinical check. You are not going to stay those blood into the water, right? You are going to put into a well. PBS under saline, right? Because there's certain iron condition on it. So blood is not going to lysis or explode it. Okay. So in other words, the iron concentration in solution is very important. If they are same equal concentration iron as the blood, we call them isotonic. If lower, we call it hypotonic. If higher, it's what? Hypertonic. If it's hypertonic, water will leak from the cell into environment. So it's lysis. It's hypotonic, the water gradient is much higher in the solution. So water will get into the red cells for maintain the same concentration of water on each side. So we use osmosis to regulate concentration iron by water, not the iron alone. Okay, that's very important concept. Another one is mentioned active transport, which require energy. Okay, and also we're against concentration compared to previous slides, previous passive transport. They were from low concentration, move to higher concentration. Okay, so need to ATP. Okay, got it as idea. Another one is a vascular transport. This is a very important part. The first one is pinocytosis. The substance is drop lipid. Okay, it's very tiny. Then stay on the cell membrane. Our cell will detect it, and they'll using the cytoplasma flow to enclose this drop lipid, eventually engulf lipid into the cytoplasm. Okay, that's what we call it pinocytosis. Another one is a receptor mediated endocytosis. It's more complicated. Only particular spot or micro domain of cells which a receptor bind to. So you mean they are more affinity, more specificity to particular stuff. When you recognize them, they will to form the pit cells, the pig structure, okay? And then engrave them and transfer into cytoplasm. Another one is phagocytosis, which responds to big molecules, big microorganisms. Okay, it's need a lot of protein involved. Sometimes need to receptor, and also need to cytoskeleton coordinate those acts. Again, from the vesicle and engulf into cytoplasm and digest in there. Okay. Give example COVID-19, right? When you use ACE2 receptor to recognize our body, right? Then get into our cells. In this case, amoeba, these protozoa animals found their nutrients then doing the phagocytosis. White cells also, okay? When they go into the injury sites, they found bacteria, they eat them, digest them in the cytoplasm. Okay, what is exocyte? Another one, the cells after metabolism, they produce a lot of waste, right? They don't need to stay in the cell, it's toxic. What happened? They need to transport out to the environment, correct? So they may be one of them use the exocytosis. Another one, again, COVID-19, after they manufacture a lot of their co-protein, their RNA, they're assembling, and in last stage, they need to use they use the exocytosis. In the end, the membrane, the vesicle will fuse with the cell membrane, and later on will release their progenies. That's we call them budding. Okay, that's another way to shedding onto the environment from the cells, infected cells. Okay, now we talk about a lot of receptor, right? Transporting thing. And the cells, especially for the animal or human, we are not single cells. Single cells did not execute anything when they became the organs, became the system. So in other words, a lot of cells need to work together with the purpose, with the particular function. So we need to work together into the organs, how to make them line it up, assembling together, and also how to regulate each other. The first one is desmosome, which is a very important structure. Okay, like a rope that use the cytoskeleton intermediate film. Okay, put the cells together. Okay, that's a desmosome structure. It's a adhesion structure. Another one we call it tight junction. Tight junction is impermeable membrane, a structure. Cell contact very tightly with a unique protein. Only allow the water and iron go through between the cells, not communicate between the cells. So go through between the gap. Okay. However, gap junction, which allows cell-to-cell communication, there is a channel that can deliver message. So there's three different structures. Put the cell into the sheet. Okay, make their contact. Okay, so now we know the basic function. Basic function components of a membrane. Now we are going next to check the difference between interior and external environment. For example, in the exterior environment, the sodium concentration may be 150. It's around 50, but in the cells, only 50 millimolar per liter. Okay, it's relatively very, very low. For potassium ion in the cell, it's much higher. Okay, and the protein in the cells is only cell own protein, so it has a lot in the cytoplasm. It's 65. You know, protein got a charge, right? Because amino acids. And so those make up make the environment difference in the charge, cell, cytosol, and the external microenvironment. Okay, so but the environment will change because metabolized product will change. So pH will change, but the cell need to maintain the iron concentration at a certain range, right? So they need to activate, they need some pump, ion pump, magnet or protein or channel to balance this environment, okay? Making homeostasis stable. Got it? So you can see that different ion concentration contributing from sodium, from potassium, so will carry the charge difference between internal membrane or external membrane. So when just consider these two ions, you will make around okay, here is potassium minus 90 and sodium plus 60, something like that. And taken together, consider other protein, other iron. We may after in the final we measure is minus 70 amp voltage, millivoltage. Okay, that's the state condition. In this condition, we call them polarized condition. Okay, and in order to maintain this condition, we need some pump exchange between them around, make it a balance. Okay, so after I told you, right, we got ion channel, we got the growth, we got the receptors. Those events can make a difference in membrane potential. Okay, the ion concentration difference. For example, in the how to say that, a state favorable condition is, I mean, stable condition, not stable. The neutral condition is minus 70 millivoltage, right? After receive the signaling, after increased ion, in this curve we call it depolarization. In other words, what increased voltage from minus 70 to achieved close, I mean, how to say that? Close, I mean, favor to the zero, we call it depolarization. At the second minute, this is 5 minutes, this is a 5-second event, okay? Then later on will decrease. In this curve, we call it repolarization, okay? And later on, they will drop into the less than 70. And this curve, we call it hyperpolarization. This kind of message is allow the neuron cells can pass their signal from cell body to the axon and to the tail and the tips of axon. Okay, and it will send a message on. Okay, then coupled with the final step, synapses. That's the reason why we got to learn membrane potential. Another case is what? It's a mitochondria, right? In the mitochondria, a lot of membrane. There are two membranes, or between them, there are two membranes, internal and external membrane. On the membrane, a respiratory chain, right? A different protein. They would pass the electron from the different position and from higher to lower. In that potential will drive ATP production in the end, will release electron, right? And pass into the end product who receive the oxygen and produce water and ATP in the end. Same is very similar to this one, okay? And how to make this depolarization or repolarization, hypolarization, there's a different type. Okay, voltage or chemical gate or mechanical gate or thermal gate. Use the heat, use the voltage, use the hormone or chemical or mechanical to make a membrane potential. to achieve bioactivity through the membrane. Okay, there are two kind of electrical signaling. Well, first one is graded potential, another one's action potential. Everyone got to learn, got to know how to distinguish grade potential and action potential. This is very important. Mostly in the neuron impulse depends on action potential, okay, because action potential can extend from one side to another side, can pass much longer. And the graded potential is transit, okay? Let's see the difference, okay? For example, you give the signal, you give the stimulus, okay? Then membrane potential will change. However, only the locally change, and it cannot express into the other area, okay? And this we call it, and this response area is we call it active area, okay? And the other region is inactive form, so it cannot spread it onto other sites, okay? And this we call them graded potential. They trigger by specific event. For example, sensory stimulus. That's a very important part. Pacemaker would regulate our heartbeat. Okay. And the next, I think this is. I think this is okay. I'm sorry that. This slide just want to say that this signal is not go far away. It's locally. Okay, you can see that after activation, right? You just nearby area with the change in the charge and get a change in the mentioned potential. Okay. And also they last very, very shortly. Okay, not stand very, very long. Okay, it's transit. And you can see that they goes just nearby. Okay, they change from -55 to -70 in the certain area only. And action potential is a little bit different. Okay, which induce higher changes in membrane potential. Each peak around the -100 minimum voltage here. And this curve we call an AKA spike, okay? And you can see that there's a figures here, we call it threshold potential here. What it does mean in this? It means if the signal cannot reach -50, this threshold potential, you are not going to activate sodium channel. You are not going to activate potassium ion channel later on. It's not good enough, not strong enough. Okay, so this threshold potential, and you can see that when you got this depolarization, I mean this stimulation here, and the membrane potential is going to change it, okay, until above the threshold potential, then will induce depolarization and activate sodium channel in this case. Okay, when open a sodium channel, what happens? A lot of sodium get into the cytoplasm site and will increase to the membrane potential to the +30. So you look at it in the static membrane potential -70. Now you go to +30, so you can reach 100 millivoltage, the difference. Okay, so that's the minima or changes, and is much bigger than gradient potential we mentioned in the previous slides. And later on, what happened? It was the sodium channel will close. How to say that word? Delay closing. Okay, after the closing up, will turn on another ion channel open as a potassium and potassium or ion channel. When you open, a lot of potassium ion will pumping out onto the environment from the cells. Okay, then will reach will drop, potential will drop it later on until to the minus 80. In this curve, from the minus 70 to curve, we call it a resting potential. Okay, so using this can generate huge impact on change in membrane potential, and also can propagate those messages into the other area and push forward much longer, and duration is much longer. Okay, that's the way in the neuron that pass the impulse from one neuron, one nerve to another nerve. Okay, that's the difference between the gradient potential and the action potential. That's very important. Okay, that's the slide I just mentioned of that. In the resting state, the sodium is not get into the channel, right? After get the receiver signaling, the ion will change the conformation, they open it up, and sodium ion is going to the cytoplasm. Okay, then later on with delay close. But in that time, sodium is getting to the cytoplasm a lot and activate potassium ion channel. Okay, use this method. Okay, conformational change to turn on two different ion voltage gate channel, sodium and potassium. And this was an all and nothing spike. I told you, right? The threshold potential is very important. If this signal, external signal, is not making good enough, more than minus 60 millivolts, what happened? Did not reach this threshold, and response is not going to with any response. Okay, there's no gate opening in sodium or potassium. Okay, your impulse, your stimulation need to over minus 60. That's very important part. After reach minus 60, you can open up first a sodium ion channel, making greater membrane potential. Later on, you can open up potassium ion channel. Okay, we call an all and nothing spike in this case. Okay, so action potential can propagate. Okay, and then in the end, from one neuron to from the cell body, right, to the tip or axon. In the end, they need to open up, I just mentioned the vesicle is very important, right, for delivery. The vesicle, in the vesicle, they contain a lot of different neuron transmitter, glutamic acid, something like that. So they need to release those vesicle in the nerve of the tip, okay, to communicate with another nerve, okay, and this connection we call it neuromuscular junction. I mean, if the neuron contact with the muscle, in this gap we call them neuromuscular junction. Let's look at it. Here's a particular neuron here. Here's a cell body as a motor neuron, which is for control the muscle contraction or the wheel, control the muscle movement. Okay, Here the cell body, which has a lot of dendrite and dendrite, which can receive a lot of information from environment or from into cells. Okay, they put the information, and then we've got a very strong axon here. And axon was engraved by different things, myelin, something like that. In the axon, contain a lot of cytoskeleton protein, like a microtubule. Microtubule is very important, which is like a railroad. Okay, for what? For carry neuron transmitters in there, enclosed in the vesicles. You know, each axon sometimes is longer than 1 meter. It's a long travel. They need something to guide it on for trafficking. So a lot of microtubule in there, and the axon and some motor protein will carry those vesicle with the neuron transmitter, sliding according to the microtubules from cell body into the tips. Okay, when they get the signaling, make a action potential, they will pass from this side to the tips of the axon. Okay, in the end, this action potential will open up calcium ion. Okay, when the calcium ion flow from external environment into the cytoplasm, what happened? Increased calcium ion will signal transduction and release neuron transmitters. And the muscle has a receptor on it, receive the neuron transmitters, and get a contraction. Okay, that's the signaling, and then we can, according to this the position of axon, we can divide into the three different parts. Here, trigger zone, and conducting zone, and output zone, which is really near to the synapses inside. Okay, and the dendrite cell body, which receive the external stimulation, we call an input zone. Okay. However, the input maybe also contain a gradient potential. They can activate it, but they did not pass the impulse of neuron from nerves to the nerve. Here's a diagram tell you how to transmit this message or propagation or the action potential. For example, here the static state, the environment is positive, a lot of sodium. Outside here is higher potassium ion, here is negative, and get insult, get the information, and what happened? The sodium will pump out. As the one sodium pump out, will change another sodium ion channel conformation and start to pump out pumping the sodium, and later on we'll close it at the delay. Close it will open the potassium ion, and potassium ion will, adjust the potassium ion and open up more potassium ion. So use it this way for one ion channel to pass to nearby ion channel, then eventually pass through to the potassium ion. Okay, use this way, like a wave, to pass the signal from one end to another end, from minus end to plus end. I forgot one thing, the axon. Here we always I think it's one here is minus end is plus end, according to the building of microtubules. Okay, or direction. And here, what is the solitary propagation? Okay, you can see that this. Okay, let's see the difference between the CNS neuron and the somatic neurons. CNS with the different part. We use the dendrocytes. And in the peripheral neurons, we use the Schwann cells, which can insert a axon, which I can provide the electronic leakage. Okay, it's isolated between environment and the cytosol, which can prolong the action potential last longer. Okay, so they can help them out to pass the message from this area to the end tip of terminal of the axon. Okay. So that I think this one I mentioned already. They send a message to the other cells, for example, your neurotransmitter, and here is the example. And what the synapse like deliver the, for example, in this case, they deliver the neurotransmitter to the muscle. Okay, and the synapse is using a lot of cytoskeleton protein like a microtubule and the actin. Okay, if you use fluorescent microscopy, you can see they are overlapping. In other words, the neurotransmitter in the vesicle will pass one tip of the axon and connect to another neurons. Okay, in this area, we call that synapse. Okay. So I may be a little bit faster regarding how to differentiate actin potential and the gradient potential. And anyway, both can regulate ion channel conformational changes that allow the ion can penetrate or through the ion channel and go into the cells or transfer the potassium out the body into microenvironment. Okay, and use this mechanism to deliver their impulse from neuron to another neurons. Okay, then later on, why this, of course, as you mentioned, cell to cell, not only rely to membrane potential and also use the growth hormone, growth factor, receptor. Okay, but in the neuron, they use action potential most the case. And the endocrine system, however, they use different methods. They use the hormone and receptor to regulate cell function. Okay, a little bit different. Okay, let's see nerve system. How many nerve systems, including, for example, CNS, that's a central nerve system, including the brain, including the peripheral, brain and spinal cord. Okay, they are CNS, as we know, and also another nerve system we call it peripheral nerve system, PNS. Okay, PNS also can divide into two parts: afferent division and afferent division. Which afferent is a sensory nerve which can feel pain and feel changes in temperature, and also relate to contraction or intestine, not intestine, okay, organs, okay. And another one, afferent division, which is a motor, is for the contraction, it can divide into two parts: somatic nerve system, autonomic nerve system. A somatic nerve system is a motor neuron. It can regulate contraction of skeletal muscle. And the autonomic nerve system, which can regulate two very important sympathetic and parasympathetic nerve system, which can regulate small muscle like a heart, okay, contraction, and exocrine gland, okay. Another unique system, enteric nerve system, which is only mediate contraction of digest organs only, okay. So that's the organization of nerve system. I know probably all you is very familiar with this part. And how many kinds of nerves, okay, can divide three parts. One part I just mentioned that is sensory, afferent neuron. Another one's afferent nerves. What's difference? The structure a little bit different here. The cell body on the sides, very long axon and divide into several parts. One part we call the central axon, which is after the cell body and close to the CNS. Okay, and also they got a receptor, sensory receptor, can feel the pain, can feel the cold, can feel the heart, something like that. And their message delivered from the part of sensory receptor to a cell body, eventually to the CNS. Afferent neurons, which is a motor neuron, which can, with the effect function, can work in on skeletal muscle, can affect organs or glands. Okay, the message delivered from CNS direction, I mean cell body to the axon terminal. That's the action body. Okay, they are two different. Another set is interferon, which is in the CNS, which can have cell body, will receive the signaling impulse from the afferent neuron. Okay, then will mediate the administrator their will or chemical into the afferent neurons for direct or control movement of skeletal muscle. So there are three different kinds of neurons. Okay, to most how say that interneuron is located in CNS. Okay, most the case, and the other two type partial in the CNS, most part in the in the peripheral nerve system. And here in the brain, in the neuron, central nervous system, with the different cells in their tissue. The first one, we all call them glia cells or neuron glia. Most of the brain tumor derive from glia cells, okay, not from neuron. We know neuron is difficult to how to say that, to divide or regenerate. It's very difficult. Okay, so most of the brain cancer derive from glia or glia cells, or called neuron glia cells. Okay, and there are four types of neuron glia cells in CNS. The first one, astrocytes. Astrocytes is very important cells which can support a nutrients, growth hormone, or exchange to the neuron cells. And another one, very important, everyone should know that, can form the blood-brain barrier. Okay, it's very important for protect. They want to make a structure to protect our CNS cells. Another one we mentioned previously, oligodendrocytes. Again, it's like a Schwann cells in peripheral neural system, which can make a solution, protect the membrane of nerves. Next one, microglia. In the beginning of the lecture, I told you that it's kind of specialized macrophage, right? Living in the brain, which you can respond infection, okay, or inflammation. Another one, epidermal cells. This cell is very important for regular flow or CSF, okay? Later on, we are going to go over it again. Here's a location here. Actually, the type cells, for example, astrocyte, oligodendrocytes, they will work together with the neuron, okay? And also, astrocyte will link with the venue or vessel in the brain, in the CNS. Here, the functions, I got the outline here. I'm going to pass it, okay? You spend the time, later on, I will send this slide to Professor, okay? And you can have it, these slides. And the base, most of the function I mentioned before already. So I'll just get by this one. As an astrocyte, which is a culture in the tech was observing in the fluorescent microscopy, and this is growing the petri dishes in vitro. Okay, how it's very easy. We have this. We use the protein which can recognize the cell surface markers, and also conjugated with the particular dye at the label with the cells or put in the intravital or put in the fluorescent microscopy. Then what happened? We give the slide source, and this dye will receive those particular wavelengths or the light source were excited, and then they don't will turn to the fluorescent dye. And we in our computer system we will receive those photon changes and we record it, and we can get an image on our monitor like this. Okay, where you can see that a lot of astrocyte here. Okay, in the yellow color, and the rest of the cell is very long tail here. I believe this is the neuron cells with long axon. Okay, now later on, there are different. I believe the four structures is very important for protect our CNS. Right? I told you, right? The neurons are in our brain or spinal cord is fragile. It's easy to get damaged, and the damage is not reversible, cannot be recovered. Not like our skin had to get cut, where healing automatically a couple days later. But nerve, no, it's not recovered. It's a lifelong damage. Okay, so the gut is very smart. They create different method, different structure to protect our cortex. The first one is a helmet. Okay, what is a helmet? Skull, right? The bone cover our brain cortex. The next one, the hard stuff, the bone is not good enough. They are going to use another cushion material. There are three, or then it's Meninges. There are three types: dura mater, arachidoid mater. Okay, another one, pia mater. Okay, the three kind of membrane attached into the bone. Okay, but it's not complete attached. We can later on we can see this structure. Okay, see the dura mater. There are two layers. Let me see it. Okay, dura mater. Okay, I think I don't like this one. That it's not clear enough. Okay, dura mater. This support two layer. So we use the next time. Okay, another one. Okay, so bone, meninges. Another one is CSF. And this is smarter way to protect our brain, our cortex. You know, our brain, our cortex, like a ball, protects, like a ball put into the ocean. Okay, got an ocean protect our cortex. So we flow on it. What is the ocean in our brain, in our CNS? That's CSF. Is a cerebral spinal fluid in there. So after we, hit something, we got a hard stuff to protect, and the ocean will protect us to prevent from the cortex to hit the hard stuff. So CSF is another way to protect our brain. Okay, another one, BBB, I mentioned already, a blood-brain barrier, which can limit the occurring of infection, right? Because the neuron injury is reversible. So God need to protect them very, very well in particular way. So if passenger in our blood it's not easy to penetrate into a neuron, central neuron. Okay, so prevent from the further damage. Okay, that's very unique protection way. The nerves, the structural venue is different in CNS is different from the other vessels. In CNS with the astrocytes, make a very tight junction. Only allow small molecule get through into the BBB and go to the cortex. Only glucose, oxygen. Okay, other stuff, nutrients or energy providing agent is not get in there. That's the reason why I encourage students to have a good breakfast, provide a lot of glucose in your blood, so allow your brain can take it in the early morning, which can help you benefit you to learn efficiently in the early morning. Okay, if you don't have the breakfast, then go to school, you don't have a fuel in your brain. Other kind of energy producing nutrients is not go through to the BBB and get into your brain. So you're not going to get the ATP energy. Okay, so it will affect your learning course without the breakfast. However, there's the limitation for BBB for drug development for the brain cancer patient. Another good drug can kill the brain tumor, but it's not accessible because the BBB. Big molecule anti-cancer drug cannot get in there. And that's the limitation. Okay. However, BBB protect you. Okay. Later on, we are going to see the structure more detail. Okay. Let's go to the meninges here. Dura mater has two layers. That's the reason why I prefer this one. Okay. You see this one layer, two layer. Another one, araconoid matter. There are two layers also. I get dura matter. You can see that there are two layers, right? You can see there's some venue go through here, sinus, okay, go through here. And beneath, between the two membrane, and the conoid matter, there are two membranes. We call them what? This space, okay, subdural space here. I'm sorry, subarachnoid space here. Okay, here the field with a lot of CS in there. Okay, and also a coin of villus which can push the blood flow and venue flow. Okay, with the one direction. Okay, that's a very important structure in these slides. And also CSF were produced in this particular ventricle. There are four ventricles. You can see the right and left ventricle and third ventricle and the fourth ventricle. There's a CSF in there. And so also I mentioned there's between the subarachnoid space, right? So you put all the cortex in there. You can see that there is the CSF. Here is the CSF. That's the reason why I called you, right? The cortex, all the brain were floating on the ocean, okay, for the protection purpose. And let's see the lateral view of brain here. You can see that here's cerebrum, here the big whole picture. Here's a brainstem here, a brainstem here, and this one is cerebellum here is okay. And the CSF, as I tell you that here another CSF inside also, and it can protect the CSF run between the meninges and the cerebellum here. So that's the reason why I tell you it's like a cushion. Allow your brain flow around, minimize the impact, okay? Minimize the injury. That's very important stuff. Some baby which develop problem, which can, okay, this is a place to produce manufacture CSF. It's supposed to put the circulation going to the whole, cranial here. If they block here, so what happened? The ventricle will take more CSF. So eventually will build up this kind of big head here. This case, we call it hydrocephalus. Okay, need to operation to drain those CSF away to reduce pressure or brain pressure. Okay, so this causes enormously enlargement of mild CSF in the brain. Okay, here is the BBB structure I mentioned before, right? For example, here is a normal blood vessel. You can see that, you can see endothelial cells. Endothelial cells, they had a gap junction, allow the molecules to go through, penetrate into the cells, okay, for easy to exchange nutrients, right? But in the brain, you can see they has another type of cells, astrocytes, protect outside of endothelial cells. So what happened? With the tight junction, only allow glucose and oxygen go into the vessel. Okay. So this structure we call them BBB, and also they okay. Now, so we just finished their four protective stuff, right? So next we're going to move on circulation on the cranial, on the brain. You can see that here. Let's start with the artery in the brain. Artery, you can see there. Okay, let's see this one. There are four arteries provide flow into the brain. Okay. Let's see this one, cardiac artery. Cardiac artery, internal cardiac artery and external cardiac artery. External cardiac artery provide the blood flow onto the face, okay, on the face. Internal cardiac artery provide the blood flow into the brain here. That's the internal cardiac artery. They are right and the left. Okay, another one is a ventricular artery here. There are two, left and right. There are two pairs major artery provide oxygen. Oxygen-rich blood from body, from lung going to the brain. Eventually, the ventricular artery, they are going to merge together at this area. This area we call it circle villus. Okay, that's a very unique structure in the circulation of artery in the brain, which you can regulate when you get a stuck or blockage on your artery or your brain. Okay, then later on, these internal cardiac artery will merge with another vein. I'm not going to name by it. Okay, last them four. Okay, then you can see that they are different name, anterior, posterior. Okay, I'm not going to go over it. Okay, last the lowest artery provide a nutrient oxygen to the brain to the cranium. Okay. And although this is very tiny structure, but however, you can see that they are per minute, they are 750 milliliters will go through into the brain. It probably roughly 50% of cardiac output. So, you know, the demand in the brain of blood is very important. People will suffer the long-term damage in the 5 minutes without a supply of the blood into the brain. Okay, so it's critical for them to use the oxygen to generate ATP. So after finish the artery, we are going to go over the venous structure here. You can see that parenchyma of the brain, you can see a lot of small venue here. And collecting the venue, they are going to go to bigger venue. In CNS, we call the bigger venue as a sinus here. There are different sinus. Okay, on the top, on the central, we've got a superior sagittal sinus here. Okay, they collect the venue, collect the blood here. Eventually, they are going to transverse sinus here. And later on, we'll go into these two venue, internal jugular vein, and drain all the unwanted or upstream poor blood into the jugular vein. Okay, that's the circulation. I just want to say one thing very important. There are two systems, two names in the venous system in the CNS. Small one we call them venue or veins, cortical veins, small one. The bigger one is called them sinus. Okay, they are all veins, just different in size only. Okay, here I mentioned already, and we got some structure to regulate the circulation, right? Again, so another structure, arachidoid granulation. This one derived from the least suprachiasmatic space here, which can direct the CSF flow. CSF flow is very important. Okay, they produce from ventricle stove layer, then with the flow going to the on the skeletal region. Okay, surface region. For example, you can see that the flow is go this one, go to skeletal sinus, for example, like this, so that they help you to make the circulation in the flow. So people sometimes, you want to see they got infection or not, will harvest the CSF to analyze you got an infection or not. Okay, in this case. Okay, so we finished the full structure for protecting CSF, CNS. Now we are going to tell the main function of CNS brain. Allow you to see, to read, right? To taste, to generate all information together, and became the knowledge, became the how to present yourself, right? And sometimes you organize, control your function, assistant work. Okay, so yeah, a lot of things they can do. Okay, and also give it a complete person for emotion, for feeling, love, hate. So it's very important about CNS, a lot of different functions on it. So the brain components can divide into 4 parts. Brainstem, okay, and cerebellum here, forebrain here, and another one, cerebellum is this part, and this is the forebrain. We should say the encephalon, okay, is in the middle part, a central part of CNS. Okay, so now later on we are going to introduce one by one, okay, by each function. When you open up the skull, right? And we can see the cortex divide into two parts, right and left. Okay, and each part gives a different spatialization. Okay, for example, on the left, it's for skill, okay, math, science, something like that, okay, calculation. On the right, it may be good in the artist work, okay, creation. So there are a little bit difference between the left hemisphere or right hemisphere. Okay, between these two parts, there are a very unique structure. We call them Corpus Callosum, this one. This is like a information superhighway, which can a lot of axon connected between the two halves to hemisphere, allow them to go get connection. Okay, you can see that there are 200 to 3 million axon projection between this callosum. Okay, it's very thick. Okay, you can see the dorsal view, you can skitter view, you can see that there's a run here. And also we can slice it, okay, and open and put under microscope. For example, this information derived from cadaver, you can see that. You can see there immediately you can image the big difference, the color in color. On the outside, you can see what? Gray and white, okay? They are different, okay? They mention that, for example, they say gray part is like a computer in our desk in the office. The white part is what? Is a cable. The cable connect another computers. Okay, so in other words, it's suggesting different function. In the white stuff, white part, white matter may be a lot of axon. Okay, and gray part may be a lot of cell body and dendrite. Okay, so the white part will connect each others. And also here is the outline or the gray matters. Because the time I'm going to skip it, and if you have time, you can read over it after that at the lecture. But I told you the basic of different function already. Okay, that's a white matter is a wire, CNS can, they come for communication. These fiber for communication connect a bit among the different computer. I mean, the computer located in different office. Okay, like a interconnect. Okay, and the cortex in the gray matters, they are divided into 6 layers. Okay, we can use a different staining to distinguish the morphology and cell type, and outlet, and they can tell which layer has what type of cells on, okay, and what kind of axon pattern. Okay, you can see that. You can see. Use it, for example, how to read these figures. A Weygert staining. You see where the stain, you can see the pattern here, the first layer, a second layer, this pattern, right? And third layer, this pattern. So you can see there are different 6 patterns. So we can layer different 6 layers in the CNS, in the cortex, okay, under different staining methodology. Okay, so let's figure out what kind of neuron, what kind of type of neuron. So this is, each neuron will work together for particular mission, for execute particular mission. Okay, and you don't need to memorize all 6 or those type of neurons or name on a layer or cortex. You has, you just know the 6th layer. That's good enough. The neurons in the 6th layer, including those type of cells, okay, and they will distribute on different layer here. Okay, now we are going to the brain. I mean the brain into the four lobes. The first one is the front lobes. Another one is the parietal lobe. Another one is the occipital lobe, and another one temporal lobe. Okay, how to distinguish them? There are two very important lines you should remember. The first one is central sulcus here. Okay, it's very important. And another one is lateral fissure here. Okay, there are two lines can help you out to distinguish to divide the lobes position. And I still emphasize, remember the first previous the slide in the beginning, tell the line on the bone or skull of animals. Plus also we do have that kind of line also in our skull, okay? And also it can help us to do the surgery, brain surgery, or remove tumor or do the treatment or stimulation, something like that. Still, but then, okay. Okay, let's see that the different lobe with the different function. Let's see the central sulcus here. That's this area here. This is very important information here. In the that's the front, right? In the here is a motor dome. It's a motor cortex. After that, by the way, is a sensory cortex. These two areas is very important. Primary sensory, primary motor cortex region. Okay, this will direct the movement. Okay, and this part is a metasensory cortex, can allow you to feel the pain, feel the temperature change, heat, cold, something like that. And later on, there's another lobe here, maybe tell you for the assistant movement, okay? And also can help you for judgment of both sides. And another area, broadcast area here, is belong to limbic system, one limbic system. Here is for speech control. It will integrate all the information to allow you to use your muscle to start to speech, and it will give the command ordering to this motor cortex, allow you to form speech with the meaning. Okay, that's a very important part, broadcast area. Okay, and the frontal lobes is for smell, and temporal lobe here is for the hearing, and this area also very close limbic system is regarding the emotion also. And brainstem is very is the center of vital life. Okay, because there are 10. I mean, we have 12 cranial venous system go to the brain, right? 10 of them will go through brainstem, so can regulate a lot of organs function for lung, right? Okay, so for breathing. Okay, so this is very important. I share another story. Okay, I told you my parents, my in the beginning I told you my father stay in the hospital for 30 days, right? In that day before he passed away, we were called three brothers. Well, I got one sister, one brother. A hospital called us back to the hospital, and he's in the critical condition now. So we went to his bed. We call his name. Surprisingly, at that time my father is coma index is at a 3. Suppose without any response at all. But he is very emotional after we call his name. Why? At that time, I keep thinking this question. He still can hear from us. And later on, I checked the textbook. I found that I told you that, right? There are 12 planning of venom through the brainstem. My father got an injury on the brainstem. Only another 2, hearing, auditory nerve, did not go through brainstem. That is the reason why he still can hear my voice, still has response. Unfortunately, it didn't work on this on brainstem. Only rely on the machine. Okay, so I'm going to use this story to tell you you can understand your structure, the function of brain, maybe help you out to explore more mystery of life. Okay, after the lecture, as I try to say. Another one, okay, another lobe, occipital lobe here, and everyone should know that, right? The visual information will integrate on this area. Occipital lobe here is primary visual area. Another region is Wernicke's area here, Wernicke's area. I'm sorry, it's here. This region will receive information from visual, from auditory here, around here, temporal lobe. Okay, then we're learning integrate, match those voice with the meaning, allow you to understanding, and also watch from the eye, know what I understand what I'm talking about. Depends on my voice, depends on through your eyes, through my movement. Okay, so this area, Wernicke's areas, allow you process to understanding people's talk while they are talking. Okay, that's a very important area. Then we'll pass the message into the broadcast area because you understand the people talking, what message they try to say, and it will process to your knowledge, and they try to respond it on this area. Okay, and this area will pass the message, signaling into the motor cortex to allow you to use your muscle on the tongue, all right, and the facial, and pronounce the meaning, pronounce the words of pronunciation, something like that, speak out. Okay, that's the whole information here. And okay, and last in the previous stage, roughly distribution, and people also divide more region. For example, in this front lobe, okay, I told you, right, this is the motor cortex, but also divide the primary and a supplementary next, and a premental cortex. Another one, prefrontal association cortex. And also there's a particular region overlapping with those regions we call it broadcast area, speech formation. Okay, I don't think you need to memorize all that, but I did spend a lot of time to try getting all the information on this slide. And you guys, if you are interested later on, you can read my handout, okay, for your information, okay. And the and this one, parietal lobe, the first one is somatosensory cortex, which you let you feel. Okay, another one, posterior parietal cortex. Okay, that's associated with them. Another area here is a Wernicke's area. I told you already. Okay, so maybe okay. Oh, everyone gonna learn this one. Later on, I'm gonna mention this one, limbic or social cortex. Later on, I'm gonna mention this one. And here's a general function, frontal parietal occipital Frontal, you can see is voluntary motor activity, speaking ability, and elaborating thought, stimulation of different area, or is primary motor cortex move different body region, again, primary on the opposite side of body. Okay, and parietal is the process sensory each region. Oh, okay, so a lot of things. And temporary is for hearing, auditory, okay, domain, the sound sensation. So it's a video. So I think you know this main function, that's good enough for you guys, because you are not medical doctor, you are not a professional, but you should have a knowledge of it. And also, like, I prepare a lot of time to generate this data for you guys. And for example, frontal was their function. Okay, basically they divide two functions. One is it's the mental basics of physical, with like a thinking, memory, problem solving. Okay, it's not actually work, it's mind work. Okay, learning, memory thing, all action on this area. Okay, frontal lobe. Another one is a motor for action. You execute, move your arm, move your leg, fingers, controlled by this motor, the frontal lobe. Okay, the motor cortex. That's the main function of frontal lobe.
Prof, I'm sorry, Prof.
Yes.
Our class time is over.
Okay, sorry, I think so. Thank you. So I'm sorry, because I prepared a lot of stuff, a long story, because I don't have a textbook. Initially, I thought just talk about the CNS. Okay, until last night I said, oh, I need to teach you. I need to carry another subject regarding the neuron transmitter thing. So I just working on last night. I prepare a lot of stuff on the CNS structure and function of it, and I know over shorter time, but it is fine. I will record it after this lecture in my office, and later on I will send my video and the film to you guys online. Right, there's a Google Class, right? I can share the file to all you guys in the video format. Okay, so sorry about that. You can see I can do a lot of, okay. Now I'm going to last slide here. I think it's very interesting stuff. Okay, I told you, right, this is a very important part for motor, for sensor. Each particular position will respond different region or your organs or your part of the body. And according to this spot, the people draw the little figures. Okay, these science figure, cortical figures here. Okay, for example, another message. This is a later view from the left side, so they can control right side or leg movement. They can feel right side, input. Okay, for example, you got an injury. You got an injury here. Maybe let's see, in around 2/3 part here. So this is sensory, right? So when we go to 2 power here, maybe around, okay, 2 power, maybe around here, maybe affect your jaws. Okay, use this mapping the position. You can feel it, or can you control it? Okay, it's very interesting stuff here. Homunculus, okay, this very interesting stuff. Okay, thank you. Should I go another one? Okay, last one. This okay. How do we image those function particular spots? How do you know this is for learning? This is for the auditory or sensor? Very easy. I told you, right? Neuron cells rely on the blood flow, glucose, oxygen. Okay, when you have certain area with a high activity, they are demanding the blood flow, demanding glucose, oxygen. So we label blood flows, particular dye on the fluorescent dye, for example. So we put the patient on the on the detector machine, right? It's sitting there. Then we talk to them. Then we can feel the high blood flow in the particular area, for example, temporal lobe. So we know this area is in charge healing. And they start to thinking and try to move something, some spot where there's a high intensity of fluorescence. Use this methodology, we can pinpoint each spot for their function. Okay, that's we use a PET image. Okay, as a methodology we use to determine the location, sensory or motor spot. Thank you. Any questions? You see, I can explain a lot of things.
Sir, may I have a question?
Sure, welcome. Go ahead.
How could short-term memory transfer to be a long-term memory? Any possibility that long or short-term memory gone?
It's a very good question. Actually I prepared this in the end of slide. Okay, the storage is different. The short term need to, long term need to accumulate a lot of short period of time. You gotta repeat it. You gotta learn it. So when you're learning, you use a different area of cortex. Okay, if short period of time, you can remember it. And let me see. Learning memory. Okay, long memory is okay. You see the long-term memory. With a mechanism duration, long short-term memory, later very much. Is as I know, they need to, okay, long-term memory you need to coordinate in different nerve tool. Okay, you should go to another cortex to process. And short-term period, the short-term memory, just go to probably one or two area. You look at it, okay, and maybe you stay for 1-2 minutes or 1 day and pass away. You did not process on it. But the long-term memory, you are going to process learning processing. Learning processing uses different lobe or brain. Okay, and you are learning. For example, now you are learning what I said, right? You are not going and you are going to memorize each single word I just repeat. You're learning in this process. You just know the concept. You learn the concept, not a single word what I say, what I post on the slides. But you want to memorize it, you need an effort. You need to process different lobe, different section. Okay, accumulate information on. So the process different area. Okay, as I try to say, because this is no other true mechanism can tell you the difference. Okay, let me see that. So for example, in the short-term memory, they mentioned that you just decoding, okay, and they did not rehearsal that much. And this one, they rehearsal also storing, and you can record it. Okay, this last cannot, just recording, but it did not store. Okay, I think this is a very good question. I will answer your question through the Google Cast, some data information to you. Actually, yesterday I checked. There's no information or material in the Google Cast. No other thing. So it's neither another access to that or not. I don't know.
Yes, Prof.
Yes. So I can, yes, I can drop the teacher material handout.
Yes. Yes, of course,
okay.
Prof.
Okay.
Yes,
I would do it right after the lecture. Okay.
Yes. Also the recording recording as well,
Okay,
Prof.
good.
We will also every meeting I try to upload the recording of our class so students can move them in long-term memory to make it persistent.
No, don't need a long-term memory, just let the concept. Okay, idea is more important for students now, for college students. Okay,
Okay, Prof.
it's more important. Kalau idea, that's good enough, I think. And
Okay.
okay, that's all my information here. Okay, this last picture is highest building in Taipei is 101, and our school spot here around here. So we very close to the center of Beijing center of Taipei. And this picture was shoot in the Xiangshan mountain. It's nearby TMU also. Maybe take the 30 minutes of, you can climb on the top on the here and take this photo. Okay. And hopefully we can meet in the Taipei Medical University in the future. Okay.
Hopefully, Prof. Thank you, Prof.
I will prepare well next time. Try to trim less content on the lecture. I'm too ambitious to prepare.
Oh, very fabulous, Prof. Very fantastic information that student I think can have from the slide.
Yeah.
Okay.
Hopefully you can learn.
Yeah, sorry for I have a problem with connection from the previous time, but I try to close my video. It runs okay now, I think. And then maybe next week, student will also discuss in Google Classroom for next week. And
I got the yesterday I got the information from Dr. Chen, right? And unfortunately, he has the duty. So he has a duty for post a PhD student qualification exam.
Okay.
He's a committee member, so he is not available next week.
Okay.
And the language thinking to switch postpone to next week. However, that's the Moon Festival in Taiwan.
Wow.
So there's class. So if you can switch another lecture into
Yes.
next week, it should be okay. Can you do that?
Yes, of course. Okay, Prof. Will be okay.
Okay, thank you very much.
Okay, thank you
any
very
questions?
much, Prof. Any other question, class?
Yes, Prof.
Okay.
I have a question.
Sure.
Is it possible to not having dominance of brain hemisphere? Can somebody develop both right and left equally?
It's not clear to me. Can you repeat it one more time? Or someone help me out? Say again.
About the dominance of the brain, Prof. Can somebody develop them all, not right brain dominance? So they are good in maths, but also good in language and art, maybe.
That's genetics.
But possible, yeah, Prof. Yeah.
Yes, you can enhance by learning, right?
Okay.
It should. So for example, my friend, right? He's a good tennis player. Unfortunately, developed a tennis elbow.
Yeah.
But he still want to enjoy the play tennis. He start to use the right hand.
Okay.
A left hand. Okay, so you still can learning your motor, motor cortex. You can learn.
Motor cortex, yes.
It still can learning, but for the science and arts, the friend, I think that's a genetics. It's very difficult. So you can balance it, but you cannot over it, your genetics, I think. But you can do the learning improve.
Okay.
Okay, that's my maybe I don't know, that's my opinion. There's no unique answer, I guess. What do you think, students?
Gimana menurut Agnia? Gitu nggak? Genetik will be depend on genetic. How do you think,
So
Agnia? Mhm.
Maybe, ma'am.
Maybe.
so you want to develop some kind of skill in the future?
Maybe you would like to be scientist as well as artist, Prof.
You just can choose one for, for leading, I guess.
Yes. Okay. Okay. Thank you very much for
Thank you very much.
the lecture today, Prof. Jungshan
Thank you very much.
Chang. Okay, hopefully we meet again next
Okay.
for the next subject or topic, Prof. Thank you
Yes.
very much, Prof.
Thank you very much. Bye-bye.
Okay. Okay, I will end this class
Okay.
today. Okay, see you again, class. Good
See you.
afternoon.
Bye-bye.
Assalamualaikum warahmatullahi wabarakatuh. Bye-bye.
Thank you,
Thank you
Prof.
so much,
Afternoon, ma'am.
Prof.
Much,
Bye.
Prof.
Thank you, ma'am.
Thank you, Prof and ma'am.
Okay, bye.